My name is John Quinn. I'm from UCC here, and I'm chairing the session today. And it gives me great pleasure to introduce our first talk of this session on foraging ecology. It's Eleanor Honan who's going to talk about snow petrels. Thank you, Eleanor.
Okay, so folks, my name is this working? Yeah. My name's Ellie, and I'm here to present some of the very preliminary results from my PhD, alongside introducing our project ANTSEA, which is the Antarctic Sea Ice Evolution from a Novel Biological Archive, which is based between Durham and the British Antarctic Survey. So, a brief overview of what is arguably the world's most beautiful seabird. That's a controversial place to say this. But snow petrels are fulmarine petrels. They're endemic to Antarctica, and they've got a circumpolar distribution. So they nest all around the continent, but unlike other seabirds, they're not restricted to coastal sites. Snow petrels can actually nest up to 400 kilometres inland because they're capable of just getting up high and covering the distance out to the coast relatively rapidly. Out of all Antarctic seabirds, especially the flying Antarctic seabirds, snow petrels are arguably the most ice-associated or ice-dependent. So if you look at the penguins, for example, you could compare them to the adelies or to the emperors, in that they're found in really close association with the pack ice and the outer margins of the sea ice zones. Compared to the other fulmarine Antarctic petrels, so they're conspecific with, they're sympatric with Antarctic petrels. They're more piscivorous, but they are a relatively understudied species. So what I'm going to present to you today is some of the first movement or fine-scale movement data that we have from this species. Now, as I think Alex mentioned in his talk this morning, some petrels will have a proventriculus, and in that proventriculus they can store food, but fulmarine petrels convert a lot of the food that they take into their stomach, into their proventriculus, into a really nutrient-rich fatty oil. So it's concentrated dietary lipids from the fish, the krill, and the squid that they're eating, and they can use this oil for chick provisioning, and also for defence. So they'll use it in arguments with conspecifics, but they'll also use it to target skuas when they come to the raped nests. And also, if you're a PhD student trying to stick a GPS device onto them, you will get spat at. So you can see here that this, out of the entire field season, was one of the few birds that actually missed me. But you can also see that this bird has spat onto the area surrounding its nest site. And this is Antarctica, so you're not going to get rain, you're not going to get the kind of weathering that you might get at an other coastal seabird's nest site. So, over millennia, what you get is these stomach oils building up around the nest sites, and they can get relatively thick. We have some deposits of these waxy stomach oils building up to around 1 metre. And these are preserved all over the colonies. So when you get to a snow petrel colony, it's not, for one thing you'll notice, it's not a loud colony because they're right down into the crevices between the boulders on areas of exposed rock. But you just have this mass of this, sometimes it looks like a soil, sometimes it looks like candle wax, draped all over the rocks surrounding the nest sites. Now, this wax has been radiocarbon dated, and this is quite a long established method in Antarctica for looking at ice sheet chronologies. When you date the basal surface of the deposits, it gives you an idea of presence or absence of the snow petrels, which can be used then to date the last time of ice sheet advance or retreat, and you get incredible ages of these deposits, so upwards of 40,000 years. Now, we have found some deposits that are slightly older, but for the most part, deposits we've recovered are around the 40,000-year-old mark. But the thing about the deposits is that they're all dietary in origin. So everything that's in these deposits, the snow petrels have taken back from the sea ice zones to their nest sites and then regurgitated or spat out. We know that snow petrels as a species are incredibly associated with the ice. And the one thing about Antarctic sea ice is that we actually don't have a good idea about it compared to, say, Arctic sea ice history. So when you look at models of past Antarctic sea ice, there's huge variation in the models. We don't have it constrained very tightly. And a reason for that is that we just don't have the coring record around the continent that you might expect from other sites. So because of the Antarctic pack ice, the cores are very patchily distributed. And what we need for Antarctic sea ice in order to better constrain our records, which will allow us to make better models to produce future sea ice range, is higher resolution archives. So you might see where I'm going with this. So the assumption that our project, well, the hypothesis that our project is making is that if these stomach oil deposits, which are the, again, a record of what the snow petrel is eating, is recording its foraging behaviour, and its foraging behaviour is intimately linked to the sea ice, maybe these deposits can in turn become a record of past sea ice conditions in Antarctica at a very, very high resolution. So this has been investigated by my PI, my supervisor, Professor Erin McClement, who leads a European Research Council grant for the ANTSI programme, under which my PhD is funded. And my part in this project, we're quite a big project, there's about 17 of us, we're very hard to avoid at conferences. My role is to constrain the habitat use of the modern snow petrels. So many people on the projects are focused firmly onto modelling past sea ice or into looking at the geochemistry of the deposits, but what I do is I look at how the modern petrels are behaving and how we can apply that to past change. So I went down to Antarctica this past austral summer, and although we were interested really in looking at overall habitat use of the snow petrels, so not just how they're using the sea ice, obviously because of the nature of the project, sea ice was a massive focus for us this year. However, if you followed the news, this year was not a normal year for Antarctica. We encountered the lowest ever recorded Antarctic sea ice minima on the 23rd of February 2022. So this is uncharted territory for the Antarctic. The snow petrels that we're tracking wouldn't have ever encountered a sea ice regime like this before. If you look at the GIFs, we're seeing that compared to the last sea ice maxima in 2015, the area around the Weddell Sea and around Dronning Maudland, that's the kind of the top bit of Antarctica as it's projected here, had far lower sea ice than ever before. But that means we can look at a really interesting question. If we have this bird that's apparently incredibly ice dependent, what do they do when there's no ice? So it's giving us a chance to eventually investigate the flexibility of the species in a way that hasn't been plausible before. So I went down to the continent in December 2021, and I was there for 5 weeks. So the aim of the project was to not just deploy tracking devices, but also to take tissue and dietary samples from the snow petrels and to collect a few more blocks of the stomach oil deposits. So we had 2 field sites for our project. Myself and Steph Prince were at Princess Elizabeth, which is the Belgian station in the Sauron-Dane Mountains, and we were able to, despite the fact that the weather was bad even for Antarctica this season, we were able to, alongside collecting dietary samples and deposits, deploy on 34 birds, and we got 29 trips back to us. My supervisor, Dr. Ewan Wakefield, was at Tor Field Station. Unfortunately, you may have seen in the press and in publications, the Antarctic petrel colony this year at Tor, at which Ewan was working, had a mass desertion, so his field season was cut short, but he did manage to get tags out on 6 birds, all of which returned a trip. So overall, from the field season, alongside all of our diet and deposit data, we got 40 trips back out of 40 deployments, which we were happy enough with given how bad the weather was and the fact that it was a COVID year. So I got out into the field a wee bit earlier than Ewan did, so I managed to capture some incubation tracks, which you can see here. These really long, sort of looping tracks out further past the shelf edge. At Svartamarin, because the tracks happened a little bit later in the breeding season, they're a little bit more constrained and more uniform as well, but you are seeing here a lot of repeatability of the same bird as well. We had PathTrack GPS devices out, and they were programmed to record a location every 30 minutes. So with these tracks, so far what we've done is we've put them into a hidden Markov model to pull out the foraging locations. Alongside looking at resting and transit, you'll see the photo I've used for the resting snow petrel. It's on water. One thing we haven't done yet is look at whether or not they're resting primarily on ice or on the open water itself. There is a vast difference between the incubation versus the chick rearing tracks, but as was mentioned in the previous talk, that needs to be taken with a pinch of salt because we only did get 3 incubation tracks out of both sites. So when you look at the distribution of the behaviours, the birds spent most of the time foraging, bearing in mind they had to travel 200 kilometres upwards to get to the sea ice, the edge of the ice sheet. They managed it for most of my points in about 4 hours. They managed to get out to the ice edge, so they're sort of fairly honking along. And we were getting a signal from the resting behaviour, a low signal, that it was occurring mainly in the middle of the day. But because of the 24-hour daylight, we weren't really going to capture a signal yet of the DL migration that you might get from other fulmarines. One thing we were really interested in is if there's no sea ice, what are they using as a guide for their foraging behaviour? And quite a strong signal that came out was that the birds were targeting certain depths. So when they reached the ice sheet, there would be some foraging that occurred over the continental shelf. Antarctica's got quite a deep continental shelf. It's about 500 metres. But then, for the majority, our foraging occurred over about the 2,000 to 3,000 metre contour. And you can see at both sites, it's really, really clustered along that depth. If we zoom in at the Svarthamaren site, so this is where you have a real concentration of foraging points. You can see that they were really localized, and also that there was no ice in this image, and this should be an area of quite high pack ice. And then this is where it got interesting. This is meant to be the most pagophilic of all Antarctic flying seabirds. This is the image you have of a snow petrel where they're foraging in the leads. This year it was more like the image below. Nearly half of our foraging points occurred in 0% sea ice cover. So if you again zoom in on the Svarthamaren site, super low. So this is for the entire tracked period. This is what we were looking at. So there's still a lot of work to be done. This is only very early data coming out. This is the first data coming out of my PhD. But hopefully over the next few years, with a few more field seasons, we're going to better constrain how the snow petrels are using their environment. And hopefully in the coming years, the sea ice regime won't be quite as extreme, and we'll get to see how the birds interact with the ice itself. So again, we're going to extend it to look at more environmental variation. We're also going to start making a habitat selection model. And then in the coming weeks, I'm going to head into the lab and start looking at the diet samples that we collected. So far, from preliminary sieving, it is just agreeing with that. Snow petrels are very piscivorous. The majority of what was spat at me was fish. And then eventually, towards the end of my PhD, we're going to bring in the modern analysis compared to what signal we're getting from the geochemistry of the deposits. So just to say thank you, especially to the wonderful Stephanie Prince, who stepped in for my field season when we had someone have to pull out, completely saved the day. Also to the International Polar Foundation and MPI for facilitating both myself and Ewan's field seasons, and for myself, the staff at Princess Elizabeth Station, especially Dr. Henri Robert, were fantastic. Our ANSI project team, especially to James Gresham and the European Research Council. So, thank you for listening, and happy to take any questions.
Thank you, Eleanor, for an excellent talk. We have time for questions.
Hi, thanks very much for that. If I remember rightly, snow petrels are quite sexually dimorphic in size. Am I right?
No, there's two morphs. So there's a lesser and a greater morph of the snow petrels.
Right.
We weren't able to pull out a difference yet, that we need to look at some genetics for that. And we did notice, though, that at our colony, the birds, and they're not meant to be sexually dimorphic, there is a big difference in the hand and in the weights, where you did feel like you were definitely dealing with some birds that were a lot more robust in comparison to others, and also markings around their eye and darker markings on the rectrices as well, but we're not sure whether or not that's related to them being either morph. Okay.
The question was going to be any indications that they were doing different things, those two morphs?
We haven't sexed them yet, so the feathers will be going in hopefully over the next few months, and then we'll be able to pull out differences. Again, we've got quite a limited number of tracks. We've only got 40, so whether or not we can pull out sex-dependent differences from that is, something we'll have to look into. But I'm really excited to do that.
Thank you.
Any other questions there? Yeah.
Oh, yeah, that was fabulous. The areas that the snow petrels were foraging in, I appreciate that you have limited data. The areas that they were foraging, would they usually be covered by ice, or would the ice sheet extend to that?
Yeah, so there would usually be a much greater distribution of ice in that region. It wouldn't be, at that time of year, it wouldn't be like full contiguous pack ice. It would be fragmented, but we would expect to see ice there, definitely.
So could it be the case that birds are foraging preferentially at certain depths, and ice is actually a constraint rather than birds being tightly associated to the ice itself?
Yeah, so that's one thing that we want to get a bit more into and look at whether or not that 2,000 to 3,000 metre contour is to do with maybe something like a feature in the bathymetry. I couldn't find any like seamounts or anything in the region. I have had a look at the new bathymetric data set that came out from IPSCO. There's not much there. It is just a shelf. But if we start looking maybe at some eddy data, we could pull that out. Did I answer that question? I don't think I did. Yeah, that's one thing we're going to pull in. And someone in the project is looking at modelling prey distribution as well.
Okay. Actually, I have a very quick question, if you don't mind. When they're foraging over the ice or in those ice crevices, it seems to me they might be a little bit vulnerable to predation in ways that they may not be at sea. And I was just wondering, do they use the fouling in those sorts of contexts, or is it only at the nest sites?
So one thing we noticed is that for all the image of a snow petrel is that they're this beautiful, pristine white seabird. After a few days on the nest, they're filthy. So they get covered, they oil each other, they get covered in guano, they get covered in the mumio. It doesn't stay below freezing all the time, so the mumio gets oily and the birds get absolutely coated in it, and they leave a lovely beige colour. When they come back, they're pristine again. So whether or not that's because they do bathe on top of the ice, they sort of roll around, it's adorable, but whether or not we're just missing the fact that they've been targeting each other out of the ice, we're not sure. I think I do have a picture somewhere, though, where it did look like it was taken at sea and they had targeted each other. Sorry, way too many animations in this. So that looks like a dispute at sea. Yeah.
Thanks very much, Anna. Excellent.
bye.
Thank you. So next up we have Sam Cox, one of our own.
Hi, so I'm going to talk to you about some work I did whilst I was a CNES fellow conducted at Marbach, which is within the ERD and CNRS of France, and it's the presentations on evidence of the use of memory, social information and wind by seabirds foraging across a tropical island ecosystem of the Atlantic Ocean. So a brief introduction. The drivers of seabird foraging movements have been fairly well studied in temperate and polar regions, where environmental productivity, seasonality, and spatial heterogeneity is strong, and we know that these have a strong influence on seabird behavior in these environments. However, our understanding of the drivers of seabird distributions in tropical regions, where environmental structure is relatively homogeneous and prey are patchily distributed in a more ephemeral way, is less developed. Here, processes relating to individual memory, particularly over short timescales, collective behavior, and the use of social information may be particularly important, whilst energetic constraints relating to local patterns, weather patterns, i.e. wind, may be present. However, investigating these processes is challenging and requires comprehensive data sets detailing the repeated movements not just of one individual, but all individuals in relation to one another. So this brings me to our study, which capitalizes on a data set of brown booby movements at Sao Pedro, Sao Paulo tropical archipelago in the tropical Atlantic. And here we had the opportunity to analyze a data set where over half the local breeding population was semi-simultaneously tracked using GPS loggers between the 8th of July and 1st of August in 2015. And specifics of this data set, combined with the characteristics of the study site and the species, present a unique opportunity, at least in my opinion, to investigate the use of memory, social information, and wind by a general seabird predator inhabiting a tropical ecosystem. So what's good about the study site for this kind of study? So first, it's a small, isolated, both genetically and geographically, population with a life history pattern that is very strongly adapted to local conditions. The figure on the right just shows you where Sao Pedro, Sao Paulo is. The nearest other bird colony or area of land is more than 400 kilometers away in Fernando de Noronha, so it's really isolated. Second, there's intense competition for nest space at the colony, so it's not a big island at all. And this means that making very short and efficient foraging trips is incredibly advantageous to ensure that you retain your nest space. Third, it's a homogeneous area. Oceanographically, it's very, very homogeneous. and it's immediately around the archipelago, and that means that we have limited habitat selection forces from the oceanographic environment. And finally, only 2 other seabirds inhabit the region in quite small numbers, the black and brown noddy, so we don't anticipate there to be strong, it's a more simplified system. And the result of this means that we get kind of a distribution that goes, it's very localized around the island, but going kind of everywhere. And trip durations are short and maximum distances are short. So this just gives us very good spatial overlap between birds. And when we couple this to our sampling protocol, we also have very good temporal overlap between birds. So over 25 days, we tracked 250 foraging trips across 95 birds. And this figure just shows you the timestamps of GPS that we have. Each row is a different bird. And so on some days we have simultaneously tracked 6 birds, sometimes 11, across a median of 8 trips, but in the best case scenario this was 23. So as I said, we then used this data to look at how processes relate to memory, collective behavior, and information from conspecifics, and wind might shape the foraging behavior of brown boobies in this tropical ecosystem. And over the next few slides, I'm just going to go through each of the analyses and the results for each of those components. So first, memory. So we wanted to ask, do individuals return to the same location from one trip to another? So to address this, we compared the departure bearings to the departure bearings of the previous trip of an individual, and we compared the distances between the centroids of the first foraging patches of one trip from an individual to, not to the next, sorry, to the previous trip of the individual. And in addition, we wanted to ask, how does any evidence of memory and repetitive individual behavior that we detect persist through time periods of greater than a trip? So for this, we compared the departure bearings of an individual to all the departure bearings that individual made across all its trips and modeled this against time between those trips. So do individuals return to the same location from one trip to another? So this plot shows our proportional distribution in difference in departure bearings between successive trips made by the same individual. So that's in yellow. Then in blue, we have results from a randomization based on 100 sampling simulations per data point, which also accounted for potential influences on wind. And if anyone wants more details about that, you can come and find me in the break. And then in the green dashed line, we have what we would expect the distribution to look like if it was completely random and birds were just going in any direction from the island. And so we see that that birds are generally departing between successive trips within about 30 degrees of one another. So there is a preference to go in the same direction as before. And this is reflected in our distances between first foraging patches. So this is the same type of figure, except this time we're looking at distances between those first foraging patches, and we see that more birds or trips than expected following a random distribution end up in foraging patches that are within a couple of kilometers of one another. And then we looked, okay, so how does this persist over time periods of greater than a trip? And we modeled the difference in angle departures between all trips and modeled that against time. And we found that as trips are further apart in time, the differences in angle bearings increase. So it's kind of like they're shifting around. And it seems to be after about 3 days, you see quite a big change in the variation in those angle departures. So this might relate to other processes to do with the prey distributions, patch depletion, or colony level behavior. So following this, we wanted to have a look a bit more about social information. So do individuals aim to forage in the same locations as recently returning conspecifics? So for this, we compared the departure bearings of an individual leaving to the return bearing of the most recent returning other bird, and we would only include paired trips in this if they occurred on the same day. So if it happened that we didn't have a record until the day before, we wouldn't compare that. And we also compared distances between the centroids of the first foraging patch of a departing bird to the final foraging patch of the most recently returning of a bird. We also wanted to look at their at-sea behavior, so do individuals alter their movements at sea in response to the behavior and distribution of conspecifics. So for this, we examined distances between foraging and traveling birds to other foraging and traveling birds, and we calculated the distance to the closest other foraging traveling bird within a 2.5-minute window of a location. And we then incorporated this information into a hidden Markov model that we developed to identify foraging locations to test if birds alter their behavior in response to the behaviors of proximate birds. So do they transition to forage when they come across other foragers? So this is the departures of all trips from the colony, colored by bird ID. The size of the marker relates to the day of departure and the position of a marker relative to the center also. So in the middle is day 1 of our study, and it goes out to day 24. And we see that we do get clustering and departure across the colony, but this does shift. So here you have all the birds are kind of coming here, then they shift around to here, then they might move back. And when we looked at the differences in departure and return bearing between paired individuals, we found, again, that the frequency proportions were higher than we would expect based on randomization, but not as closely tied as they were when we looked at successive trips from the same individual. So there we had a lot more within about 30 degrees. Here it spans out to about 70. It's also worth noting that you don't get many birds in both scenarios doing something completely different to what they did before or the rest of the colony did before. So it's very rare that you get angled departures of more than 90 to 120. And this was also reflected in our distances between first and last foraging patches. So we found that generally it wasn't as closely as coupled, but that they were going to the same area as where birds had returned from. We then looked at the at-sea behaviors in response to the behavior and distribution of conspecifics. So these graphs show the frequency proportions of foraging traveling birds within 600 meters of other foraging traveling birds with relative time into the trip. So if we take the first figure, this shows us the proportion of foraging birds that are within 600 meters of other foraging birds with relative time into the trip. And this grey line is what we would expect from a randomization, and the red is what we observed. And so we found that foraging birds were not particularly clustered at the beginning of a trip, but during the central component of the trip, they were very clustered, and then they kind of spread out. For travel to traveling birds, we found the inverse. So birds are leaving the colony, they're a bit closer to each other than we would expect from random. And then when they get out to sea, it's like they disperse. So they don't tend to cluster together when they're searching for food. They seem to spread out a bit. And as they come back into the colony, they come back in as they come into a smaller space. And if we look at the distribution of traveling to foraging birds, we find that it's rare that you find a bird traveling when it's close to a forager, because generally they will transition into foraging behaviors. And our results from the hidden Markov model reiterate that. So this is the transition probabilities with distance to other foraging bird. And we see that if a bird is traveling, when it is close to a foraging bird, it will transition into a foraging behavior. And this effect is much stronger when the forager is closer. So this is at 200 meters, 4, 6, 8, and so on. So finally, I better rush actually, we looked at wind influences and how they might constrain energetics. So we looked at differences between the bearings of departure and return of birds in relation to wind direction, and we assessed if there were any preferences towards headwind, crosswinds, or tailwinds. And here, very quickly, a difference of 0 would say the bird is flying into the wind, so headwind, and 180 is a tailwind. We found in departures we had a very slight preference to a crosswind, whereas in returns it was fairly uniform. But when we looked at this in relation to wind speed, we found that at stronger wind speeds, birds would orientate themselves to take account of a tailwind, and at lesser wind speeds, they will fly into a headwind. And we found that consistently between departing and returning. So in summary, Together, our observations suggest memory, social information, and wind do play an important role in shaping the foraging strategies of brown boobies at Sao Paulo, Sao Pedro. From one trip to another, individuals forage in similar areas to those visited previously over a time period of around 3 days. Individuals also leave the colony and forage in similar areas to those of recently returning other birds. When at sea, foraging birds are clustered in their distribution, whilst travelling birds tend to be more dispersed, particularly during the central section of a trip. And travelling birds transition to foraging behaviours when in proximity to other foraging birds. And finally, birds do seem to adjust their behaviour in response to changing wind conditions, possibly due to associated energetics. Thank you, and sorry.
Great talk.
Sorry,
Thank
I
you, Sam.
ran
No, I don't. I think we've time for questions. So, questions?
Sorry, I forgot to say thanks to the collaborators and funders as well, especially Guillaume.
Middle of the room there. Thank you.
Hiya. I'm just curious, I don't know anything about brown boobies. Do you know if there's any evidence to suggest that they find prey patches using olfactory cues? And if they do, whether you'd be able to disentangle sort of information use from conspecifics from just a common driver of where prey patches are, so thinking of the angle of that they are leaving the colony from.
I don't know specifically for brown boobies, but they're in the Sula family, which is very closely related to gannets. And my understanding of gannets is they have fused nostrils and a very small olfactory bulb. So I don't think it's anticipated that smell is a big component for them. In the case of Al's study as well, it's a really small area, so I'm not sure how sensitive the smell would be that they would be able to. I always imagine that if you're using olfactory cues, it's maybe on a slightly bigger spatial scale than the fine scales that we're looking at. If they were, I think it would be very difficult to disentangle that, and that's what was nice about our study system, was that it was very homogeneous. I've done a lot of analysis looking at the local oceanography and trying to link that, and you don't find patterns between local oceanographic conditions and what the birds are doing.
Thank you very much. We have actually run out of time, so another round of applause. Thank you very much, Sam. Next up we have Lizzie Permain, or is it Permain? Permain, who's going to talk about foraging behaviour in wandering albatrosses.
Right. Good morning, everybody. My name is Lizzie Permain, and I am a first-year PhD student based at BAS, and I'm going to show, this is very much a work in progress, but some of my preliminary results, and I'm interested in the drivers of foraging distributions and behaviour in wandering albatrosses during the breeding season. First of all, just to thank my supervisors, Richard and Andrea, and Anna at BirdLife, who coordinated a lot of the data collection for this project. Firstly, to introduce the species and the study site, wandering albatrosses are a very long-lived, very slowly reproducing seabird species. They breed at various islands around the sub-Antarctic. Here we're looking at Bird Island, which is in the very tip of South Georgia, down in the South Atlantic, and it holds about 15-20% of the global population. Wandering albatrosses are biennial breeders, so adults arrive to the colony in November, eggs are laid in December, and incubated through till the chicks hatch in about March. When the chicks are very small, they have to be brooded for about a month or so, and then after that, the post-brood period, the chicks can be left alone at the nest, and the adults can leave again. And the chicks then fledge the following November or December. So the breeding cycle takes a full year, which means that if the adults are successful or if they fail relatively late in chick rearing, they then take a year off, and that's called the sabbatical period. During the breeding season, the adults are therefore acting as central place foragers. During incubation, one adult is always on the nest and the other adult can leave. During brood guard, similarly, one adult was brooding the chick and the chick has to be fed quite often. And then during post-brood, both adults can leave and the chick can be left at the nest. However, in the sabbatical period, the distribution is much bigger. Some sabbatical birds head off, they disperse widely around the Southern Ocean. but the individuals have variable distributions during the sabbatical period. As well as breeding adults, other life history stages include immatures, so there's a very long immature period in this species. And we don't know very much about what they're doing. They head off to sea for up to 5-6 years or so. They do then start to return to the colony for increasing amounts of time every year to find a mate. They kind of hang around dancing at each other. They might defend a nest site, then they pair, and the age of first breeding is around about 10 to 11 years on average. With these sabbatical adults, as I mentioned, the successful breeders often leave and disperse quite widely, but we do get sabbaticals attending the colony during the breeding season, and there's very little divorce in this population, but they might be attending the colony and not breeding if they do need to repair or if the partner hasn't returned for some reason. Again, there's very high individual variation, particularly in the age of first breeding. It can range from 7 to 12 years or even later. And there's also quite high individual variation in breeding frequency. Some birds will defer for multiple years at a time. Some birds will breed every other year. And I should mention that birds that fail early in incubation or early chick rearing will often breed again the following year. So I wanted to look at the drivers of different strategies during the breeding season. So hypotheses include the central place constraint with the adult breeders. That changes over the course of the breeding cycle, as I mentioned. Secondly, wandering albatrosses are a very sexually dimorphic species. Males are about 20% bigger than females. This is the case in quite a few albatross species, and it generally results in males foraging at higher latitudes, maybe because they've got higher wing loadings, so more windy areas are more optimal for them in their flight behavior. Finally, I want to look at those non-breeding life stages, so the immatures and sabbaticals. Do they have lower requirements and constraints when they're attending the colony, and how does that affect their behavior? Do they disperse more widely? Do they maybe prospect other colonies? The tracking data that we've got were collected in 2 breeding seasons from breeding adults in all the stages, sabbaticals and immatures. We used a combination of GPS devices that had to be collected and satellite tags that didn't have to be collected. And initially, we used the satellite tags on the immatures, but we kind of realized they were coming back to the colony, so we started to deploy GPS devices on them as well. That might become relevant later. I should mention that these are just the sabbaticals that are attending the colony, not the sabbaticals that have dispersed and have left South Georgia. We got about 350 trips from 190 individuals with a wide spread of ages, and we also deployed geolocator immersion loggers to test when they're in the water and when they're not. Firstly, let's look at the breeding adults. We've got some trip maps here with the females in yellow and the males in purple, and a couple of summary statistics, the trip duration, and the maximum distance from the colony that the birds get to in their trips. These patterns are pretty much as expected. During incubation, when they're leaving their partner on the nest, that partner can be there for a couple of weeks, so the birds can disperse a lot more widely. During brood, that tiny chick has got to be fed every few days, so the distributions are much more constrained. And then during post-brood, as the fasting capacity of the chick increases, the trips can get a bit longer again. Now to look at the sex differences, we see really striking sexual segregation in this species, particularly during incubation and later in post-brood. As expected, and similarly to other albatross species, the females are using areas further north and the males are using areas further south. And that can be seen in both the core and general use areas and also the latitude at that maximum distance from the colony. Interestingly, this contributes to different rates of bycatch. We see higher rates of bycatch of female wandering albatrosses in tuna fisheries in the subtropical waters, and formerly high bycatch rates of males in the Patagonian toothfish fisheries further south around Birdwood Bank in South Georgia. Finally, let's look at those non-breeding stages, the immatures and sabbaticals, and I put the incubation map up there just to compare. They are using pretty similar areas to the adults, and we don't really see much evidence of them dispersing more widely or using different areas, so maybe that assumption that they're not operating under a central place constraint might not be fully justified. We do still see sexual segregation, particularly in the sabbatical adults. Also in the immatures, but it's a bit less strong, so that suggests to me that maybe the immatures are a bit smaller than adults. They haven't quite reached the same mass as adults, so maybe that size dimorphism isn't quite as pronounced in the immatures. I'm still looking into that. And finally, a little bit more on what the immatures and sabbaticals do. One of the things that we notice is they do a lot of these small trips. I've got a couple of examples here with the points coloured by the data from those immersion loggers on the bird's legs. So the orangey red points are when the bird is dry, so it's probably flying, and the blue points are when it's wet. They do a lot of these trips where they sort of head out from the colony, sit on the water, drift for a bit, and then come back. And we don't really see the adults doing that, so maybe it's just because the immatures and sabbaticals have got a lot of time on their hands. We don't really know what they're doing, but they might be displaying to each other while they're out there on the water as well. And we didn't see any evidence of them prospecting other colonies, which we might have expected to see. A little bit more on that immersion data from those geolocator immersion loggers. We can use some of this data as a proxy for foraging effort. So number of landings per hour in the daylight, which is when they're doing most of their foraging. And we see that that is highest in the 3 adult breeding stages that are provisioning chicks and brood and post-brood, and lowest in the sabbaticals and the immatures. So they're not spending as much effort on foraging. So just to summarize, with the breeding adults, we see a really clear effect of the breeding stage on their distributions and their behaviors, and a really strong effect of sex. And what I want to look at next is we do have the age of most of these birds as well, because this is a really long-term study colony. And we have seen some effects of age, even just in adults. So far, I've pooled all of the adults according to age. In the Indian Ocean colonies of wandering albatrosses, we see that males forage progressively further south as they age, but that hasn't been seen in the wanderers in the South Atlantic. So I want to look at that and see if there's any patterns by age. Finally, for the non-breeders, we see that the immatures and sabbaticals that we have tracked were acting as central place foragers, so clearly there's some sort of constraint on them there. Again, I'd like to look at the older and the younger birds to see if the younger birds are more variable in their foraging strategies. And with the older sabbaticals, are they segregating from the breeders even within that area? Are they using slightly different habitats, or are their foraging behaviors different from the breeders? Again, just to note, this is the sabbaticals that are attending the colony. Obviously, the sabbaticals that have dispersed widely away from South Georgia are very segregated from the breeders because they're used in completely different areas of ocean. And I also want to look at why those sabbaticals are at the colony. What's their breeding history? Why are they at the colony and not breeding? Maybe their partner has died, maybe they're repairing. And finally, for the immatures, why are they attending the colony? There's sort of 2 schools of thought here. Are they developing their foraging skills, learning good areas to forage around the colony? Or are they primarily concentrating on dancing at each other, finding a mate, and defending a nest site at the colony? Given that we see quite low foraging effort in the immatures, maybe that suggests that the latter is their priority. But again, I want to look at their age and how long they've been coming back to the colony to try and work out how their foraging behavior is developing in relation to the adults. All that remains is to thank Anna and the field workers at Bird Island for collecting this data over the last 2 seasons and then previous decades. And thank you to the conference organisers for such a great conference.
Thank you, Lizzie, for an excellent talk. We have time for questions.
Hi, Lizzie. Thanks very much. That was a really lovely talk. Just wondering if you know if there's any difference in the body condition or the breeding success between the sabbaticals that remain near the colony or those that travel far and wide.
That is exactly what I want to look at, because the hypothesis would be if they're successful adults, if they're good quality individuals and they're successful in breeding, they will have left. They won't be at the colony during the breeding season. And there's the hypothesis is that individual quality is what drives this difference in in whether an adult is deferring for multiple years at a time or breeding every other year. So yeah, that's exactly what I want to look at, the breeding history of these individuals. We do get some birds that just don't breed for many years at a time. So maybe that's the ones that we're seeing at the colony. So I'm really excited to look into that resighting data, the breeding history data, and get to grips with that.
Thank you.
I'm sorry if I missed this, but do you see differences in the so-called true sabbaticals that have been successful and have a full year off, and the failed breeders that could potentially depart for a period of time but start returning in the season that they failed?
Differences in terms of the actual distribution?
In terms of their colony attendance and potentially some of these histories that you're interested in?
Well, yeah, so failed breeders, if they fail early, as I mentioned, will leave, and then they might come back the following year and breed again. There are differences in between individuals in whether they fail kind of multiple times. Breeding success is generally really high, but there are individuals that do fail more. So that's exactly what I want to look at, is these individuals that are coming back to the colony. What is driving them to come back to the colony? Why are they here and not breeding?
Thanks for that. But the failed breeders, do they always go for the rest of the year, do they? They only return the following season, they don't?
Yeah, they generally leave.
Yeah,
they leave the colony. Yeah,
that's great.
Exactly. Yeah, they don't hang around after they fail. They take a year off. And they might then take another year off, but they usually will come back and breed again if they've got enough time to recover.
Okay, if we've no more questions, I think we move on. Thanks very much, Susan. So next up we have Jamie Darby, who's going to talk about underwater visibility in Manxire waters.
Hi everyone, my name is Jamie Darby, and I'm going to be talking about some work that was conducted right here in UCC. And everyone on this paper is currently in the audience, which is quite nice. So I'm going to talk about how underwater visibility constrains the foraging behaviour of a pelagic seabird. And we are, of course, talking about the Manxire waters you saw from the first slide. So Manxire waters can dive down to 50 metres for food, which I think was quite a surprise when it was first discovered. We can tell from the layout of their eyes that they have this binocular vision centred around the bill. So the placement of their eyes suggests that they rely on visual stimuli for prey capture, as you can see from the figure on the right. So light levels are clearly important for foraging. Almost all their dives occur during daylight hours. And many pelagic seabirds, these included, are likely to use visual cues for foraging both above and below the water. So the Manxire water is an ideal model species to investigate how environmental conditions affect visual hunting at sea. And why is this? They have a really big three-dimensional range. They cover this huge expanse of continental shelf, and they also cover quite a bit of the water column, so vertically as well when they're diving down to catch prey. Oops. So turbidity is the measurement of light transmissibility through a fluid, directly proportionate to visibility in the water column. Traditionally, measuring visibility was done using a Secchi disk depth, or Secchi disk depth, depending on how you want to pronounce it. I'm not sure which is correct. And this involved going to the site and sampling the visibility by dropping this disk into the water and seeing when it disappeared. Now, this required direct sampling, and some of that direct sampling was extremely useful over the years. But more recently, Secchi disk depth can be modelled by measuring the frequency of light emitted from the ocean surface. So depending on the frequencies coming back, you can tell how far the light penetrated before it was reflected, which is really useful. And many studies have used a combination of traditionally on-site sampled Secchi disk depth and satellite-derived Secchi disk depths to show that there's this huge increase in turbidity over continental shelf areas that are really important for seabirds, especially the Manxire waters. So a lot of these were focused on, say, the continental shelf around Ireland and the UK. So you can see here on the right that visibility is decreasing over the last century or so. So the working hypothesis here is that Manxire waters' foraging and diving behaviour is constrained by underwater visibility. The aims of this study, to collect and analyse location and dive data in tandem. We are going to append descriptors of underwater visibility to dives and track points, and we're going to test the effect of visibility or turbidity at 3 different scales. So broad scale above water search behaviour, so when they're in flight looking for visual stimuli or other stimuli to begin foraging. Then we're going to look at how dive frequency is affected by underwater visibility, so how often are they diving at any given time. And then we're going to look at dive depth according to these variables that describe underwater visibility and how far they might be able to see underwater. So first off, tagging was carried out on a little salty island in Ireland, in the southeast right here. We used these Pat-Track 3.5 gram tags, which are really nice and tidy. We also used some slightly bit, I don't know why it's skipping forward. We also used some slightly bulkier GPS tags in tandem with Cefas TDRs. Tags recorded location every 5 minutes and then depth every 2 seconds when underwater. So the location and timing of dives, we have this nice spread of tracks going along the south of Ireland and up into the Irish Sea. So I'm going slightly offshore. This is actually a little bit atypical for Manx shearwaters. Usually they mix their tracks between short, distant tracks and then going way out over the edge of the continental shelf to the west. But anyway, this is what we're working with. We have 36 individuals and 79 trips. And all of the dives pretty much take place between sunrise and sunset. And we had 5,472 dives to work with. And this is a typical kind of dive profile of depth over time. So this one's quite straightforward, so straight down to 40 meters and then back up again. Environmental variables then. We used model SESI, this depth that I described earlier, to describe oceanic turbidity. This is sourced from Copernicus. We use cloud cover as well, as this is likely to impact how much light is available at the surface of the water. Solar angle then was based on location and time, so given location on Earth and the time of the location fix, we could calculate what angle the sun was hitting our sunlight was hitting the water. And then seabed depth was also taken from the MARMAC package, and that queries the GEBCO database. So first off, broad-scale search behavior. So behaviors were modeled using three-state hidden Markov models, which you'll have come across. These model states based on movement phases alone using step length and turning angle. And basically the middle one of these, the middle state, where the distances are quite short and turning angle is steep, we're assuming that birds are engaged in search behavior, and that's likely to correlate with foraging. We found that foraging was much more likely over areas of clearer water than over areas of turbid water. So this is excluding dives completely. They're more likely to search and engage in searching behavior in clearer waters. Dive frequency then. So we used GAMS to study this, and very basically we were looking at how different environmental variables relate to the dive rate. So we found that dive rates were way higher during the day, which is what you'd expect by looking at the graph here, saying that all dives took place during the day, so solar angle was very important. But also turbidity. So moderate levels of turbidity were likely to lead to higher rates of diving compared to when the water was way murkier. And also time of day was quite important, and we had a peak later in the day for diving. Then we looked at dive depth, and this is my favorite one aspect of the modeling that we undertook. We again used the GAM using similar covariates, and solar angle together with turbidity was likely to be the most effective predictor of dive depth of these birds. So in occasions where the solar angle was high and turbidity was low, which meant that water visibility was very good, they were likely to dive quite a lot deeper. Clear skies also led to deeper dives, further reinforcing the idea that light levels are limiting their functional dive depth. The conclusions from this then, that Manx shearwater foraging behavior is impacted by underwater visibility. They might not dive as deep when they can't see further underwater, which makes sense. It's a really nice, simple story. Dives are deeper in clearer waters and in low cloud cover, suggesting that prey capture might be facilitated by water clarity, which makes sense given the placement of their eyes and that they're likely a visual forager. Broad-scale search behavior is also impacted by turbidity, so it's likely that the visual cues that they're using above the water are also constrained by this feature. And oceanic turbidity is increasing over vast areas. This is in part due to climate change, so the processes that drive oceanic turbidity include kind of wave action, resuspension of sediment, and this is all becoming more extreme as climate change progresses. And this may also apply to other marine top predators, so it's something that's worth bearing in mind when looking at how environmental variables will affect your study species, responses, and behavior. So, with that, massive thanks to all the wonderful co-authors, to everyone who helped out on fieldwork, and finally a huge thank you to all of you, and especially everyone who gave me their talk on time. That's all.
Thank you very much, Jamie. So we have time for questions.
Hi, Jamie. Thanks for that. That was some really cool stuff. I was wondering if you considered how much that like diurnal vertical migration might potentially be affecting that. So, like in the middle of the day when solar angle is really high, you might have, I guess, the sprats or sand eels are essentially diving deeper, contributing to the birds having to dive deeper as well.
Yeah, so location of prey is kind of a big variable, and there's a lot of unexplained variation in our models, which is likely due to the different, the changing dial patterns of prey availability. But also dive frequency and also the broad-scale search behavior is highly correlated with these factors as well. So we think that we can use that to say that it's not just where the prey is in the water column. If the prey was all really accessible in times of, poor water clarity, then surely the dive rate and the search behavior would also reflect that. They'd probably be searching a bit more intensely and diving a bit more frequently, but that's actually not the case. So in this case, this is why we use the 3 different scales. We look at the broad scale, medium scale, and fine scale. So not just the dive depth, because as you say, that might just reflect where the prey is in the water column and not necessarily the factors, driving their prey capture abilities, we'll say. Anybody else?
Yeah, thanks. So I've just got a question about the bigger significance of this. So I was wondering whether you've been able to explore whether any events that perturb the ocean bed overlap with foraging areas and lead to a reduction in breeding success at all.
So some of the examples that kind of set us off on this study were looking at planktonic blooms and how those sometimes led to mass die-offs. So like huge areas of sea being covered by coccolithophores, say in the Bering Sea for the bloom in the early 2000s, that I think it was, led to this huge die-off of a different shearwater species, and that was thought to be because, over a huge area, all of a sudden water visibility was decreased. Also the prey shifted their distribution again, but we think the combination of both of these things, this huge area that all of a sudden the visibility was very poor for, we think that contributed to their starvation, and then they washed ashore emaciated. So that was another kind of signal that this might be a really important thing to think about. And in terms of storm events as well, it also kind of casts a lot of light on why they might be starving in storms. So like Emily's slides yesterday showing that a lot of their shearwater species were turning up emaciated, it might be a problem with visibility or even just the mechanics of feeding, of diving during these storm events. So yeah, certainly it kind of starts to explain some of these things as well, which is really nice.
Yeah, cool, thank you.
Thank you.
I have a quick question, actually. When we think about these huge great balls of fish out in the sea, we often see them in pristine bodies of water. I was just wondering what's known generally about how turbidity influences these kinds of species of prey in terms of distribution. Are there any general patterns that people have found?
Yeah, so Dag Axnes, who's done a lot of work on turbidity at really broad scales, and especially how it affects kind of commercial fish species distribution, found a massive correlation between turbidity and the distribution of these species. So positive correlation with visibility and the biomass available for marine top predators. In his case, he was looking at commercial fisheries first, but a byproduct of that, I think, was just the biomass of small to medium-sized forage fish in general. And it was a really strong relationship. So it's likely that it actually affects, multiple steps of the trophic level, trophic chain. So yeah, that's kind of the only example I can think of off the top of my head. But besides him, it's quite understudied really. So it'd be nice just to think about how the distribution of other species is related to this as well.
Thanks very much. Thanks again, Jamie. I think we move on to the next thought. Thank you very much.
Thank you.
Yeah. So next up we have Fernando Medrano, who's going to talk about the white-faced storm petrel. Thank you.
Thank you all for attending to this talk. In this talk, I'm going to show you the very preliminary results on the research that we are doing, trying to look which are the intrinsic and extrinsic factors that could be driving the foraging trips during the breeding season in the white-faced storm petrel in Cabo Verde, in Fernando Medrano. And this work is led by Constanza Rivas, who did her master in the Seabird Ecology Lab of the University of Barcelona. So as a context, the habitat selection in general is a key for planning the conservation of oceanic predators, but this is particularly challenging in tropical oceans since the resources are patchily distributed and are hardly predictable. In the particular case of storm petrels, there is a recent study due to the miniaturization of the tracking devices that found that the European storm petrels in the Mediterranean select for static features such as shallow areas, but also some dynamic conditions such as water mixing and areas with particularly rich in primary productivity. In the case of the white-faced storm petrel, this species is distributed in the tropical and subtropical oceans all over the world, in the Pacific, Atlantic, and Indic Oceans. And little is known about the foraging ecology, but a recent paper in the Cervallen Islands found that they use open waters. So the aim of this talk and of this research is identify which are the intrinsic and extrinsic factors that might be driving the habitat selection of this species. So our fieldwork was conducted in Cabo Verde, in Western Africa, in particularly in Ilhéu dos Pásaros, which is this island here, which is mainly a sandy beach, where the storm petrels are the only seabird in this particular islet, and they dig their burrows in the sand. Here we installed 100 nest boxes, and we followed them every day during the breeding season, and we deployed some PathTrack NanoFix mini tags, which weighs in general more or less the 2% of the weight of the birds, and we deployed them in the central rectrices. So we obtained 126 complete trips from 96 GPS recoveries in 4 years, from 2019 to 2022. Here I wrote 2018, but it's wrong. We pre-processed the data for avoiding errors and for cutting the trips of the deployments in individual trips using the Track2KVA package in R. And we also did a matrix of cell absences for trying to understand the habitat selection by rotating the real trips in up to 180 degrees, because all of the birds, as I show you here, are going to the east of the colony. So we only rotated them in the eastern direction. And we used some intrinsic variables that could be explaining the foraging trips, such as if the trips were short or long. And for doing so, we clustered them in short trips and long trips using the trip duration of the whole trip, the distance covered in the trip, and the maximum distance that was reached from the colony. Since the breeding stage and the sex could also be influencing on the on the foraging trip and the habitat selection. We also sexed the birds molecularly, and we indicated in the models if they were incubating or chick-rearing. From each of the points, or from each of the locations, both the real and the simulated locations, we extracted some environmental variables, such as the primary productivity, the sea surface temperature, and the salinity, since the archipelago is under the influence of the Canary Current. Also, we extracted some information on the sea movement and water mixing through the FSLE index and the sea level anomalies that indicates the vertical and horizontal water mixing. We also extracted the wind speed, since the bird movement might be mediated by the wind landscapes. And since for planning the conservation, probably it's more easy to look for static features, we also looked for if they were selecting for a particular seafloor depth and the seafloor slope. And finally, we also looked if they were visiting some seamounts, since Cabo Verde is a volcanic archipelago, so there are several seamounts in the surroundings, and preliminarily, as is indicated here, there were several trips and several locations that were visiting the Senghor seamount. So we built several GAM models with all the intrinsic and extrinsic variables as fixed effects, excepting by the year, the trip ID, and the bird ID as random effects. And we did a backwise selection based on the ADF values of the GAM and the AIC criteria. And after this selection, the best model was this one. And in the case of the locations, of the real locations that visited the seamounts, we built a GLM for understanding under which conditions the seamounts were selected. So, unsurprisingly, the trips during the incubation were much longer, like it happened in the Sevagen Island for the same species and for most of the tube noses. And this is the general result of the model. So the birds, both during the incubation and the chick rearing, are selecting for waters that are colder, that are less saline, and with a lower with a higher sea level anomaly. I'm sorry, I didn't explain very well how to interpret this. But the zero value means that the trip was a simulated trip, and the value 1 means that are the real trips. So when the curves are descending, is because the birds were selecting the lower values. Surprisingly, we found out that in the case of the chlorophyll, the birds selected in the case of the chick rearing, they selected for areas with less chlorophyll, but this could be explained by the fact that actually the birds don't eat phytoplankton. So probably these birds are, there is certain lag between the moment and the places where there is more chlorophyll concentration, and when the birds are actually visiting when there is more concentration of zooplankton. Also, in the case of the seamount selection, the seamounts were only selected in years with general low sea surface temperature, salinity, and productivity, and with more water mixing. And this is seen in when we were building the kernel contours of 2019, for example, in which in this year the general system was less productive, and several birds visited the seamount here. It's even included in the kernel contour of the 20% of the density, while in richer years, like 2021, the birds were selecting waters much closer to the continental shelf in front of Senegal. So as preliminary conclusions, the incubating birds conducted a higher proportion of long trips than chick-rearing birds, as was described. And contrasting with the conclusions for Selvagem, who inferred that there were no selection of seamounts and of the upwelling, our birds, especially during incubation, traveled to the upwelling, while during the chick-rearing, the birds were constrained to the surroundings of the colony. Also, the incubating birds selected some static features, such as the seafloor depth and the proximity to seamounts, especially in low productivity years, while in chick-rearing, the birds are constrained to the surroundings of the colony and only selected some dynamic features. So planning their conservation during the chick-rearing is much more challenging. And the habitat selection could be determined by the yearly productivity of the canary current, which challenges planning the conservation of this species. So the next steps could be, for example, to check out the habitat selection in other tropical seabirds in Cabo Verde, such as the Cabo Verde stone petrel, to detail the influence of the wind in the navigation of the small seabirds, because we only worked here with the wind speed, but not with the wind angle, which could be crucial in the habitat selection. And we are also expecting to be able to compare those the diet through DNA meta-coding between those trips that actually visited seamounts against those that did not visit seamounts. So thanks for attending, and thanks for the funding of all the organizations, especially to the ANID, who funded my PhD, and to Erasmus, who funded the Master of Constanza. Thank you.
Thank you very much. Thanks for a great talk. We have time for a couple of questions. One at the back here and one at the back.
Thanks. Super work, Fernando, and really interesting to see the long and short foraging trips. In this system, which doesn't seem to be the case in all storm petrel species, in this system, are those long and short trips alternated? And how do you think that relates to the size of meals delivered to chicks?
Yeah, apparently the birds are less limited in a way during the incubation, since they conduct more oftenly long trips, despite sometimes during the chick-rearing they also conduct the long trips because they have that dual foraging strategy. While during the chick-rearing, when they conduct more short trips, or even the last days of the incubation, they are quite constrained to the area of the surrounding of Cabo Verde. So they have, apparently, they have less to select. So we are heading to compare actually the diet as well through DNA meta-coding, and we expect to do this in the next year. So, thank you.
Hi, thanks very much. That was really interesting. I was just wondering, when you were selecting your pseudo-absences, you said you only went one way because the birds always went that way. Do you know why the birds always went east instead? Was it east instead of west, I think?
Yeah, we don't really know, actually, because the wind is coming from the north. So it's not because they are avoiding the wind, actually. We don't expect that it's also because of competition, since, I don't know, I had troubles imagining the storm petrels competing like by interference, for example. Maybe they could be competing by exploitation, but yeah, we don't know, actually, unfortunately, but all the trips are in that direction, so it's very important. It's very interesting. It might be mediated by the fact that the Canary Current is in that direction, as I shown in one of the in this one. So probably there is more productivity than in the west. But thank you.
The last talk of this session by Christopher Pollock, who will be talking about foraging site fidelity in gannets.
Hello, everyone. Today I'm going to talk about some work I did during my PhD at the University of Leeds using individual-based models to try and understand the emergence of foraging site fidelity and what decisions are being made. So as a brief introduction to kind of what individual-based models are, we heard some great stuff from Emily yesterday in the plenary. I want to take a bit of a deeper dive. So they're defined as the modeling of complex systems through autonomous entities or individuals, also known as agents. And the key thing I want to highlight is these kind of emergence or emergent properties that you get. So this is an example on the right here from a very early agent-based model. And each of these dots or individuals will have 3 rules for cohesion, alignment, and separation. And the swarming that you see kind of emerging then is not actually programmed in. That's kind of just happening from these 3 rules. So you can see instantly why you potentially get interesting things for movement, ecology, so on through that. They're also commonly used in management and conservation applications. One kind of relevant example is the Seaboard model developed by UKCEH, which looks at kind of the impacts of offshore wind farms on individuals through displacement and avoidance. But they're also pretty useful for developing theory and kind of looking at these fingerprints or emerging things and trying to understand what might be underneath, and that's what I want to do with foraging site fidelity. So to recap on that, it's very well understood in the study species gannets. There's been a lot of kind of classic papers. And it has these characteristic patterns which are useful for us, which is this fingerprint of repeatable departure angles within individuals, overlapping foraging areas, and it's also not absolute. So, I mean, there's variation between individuals, and you can quantify it and put a number on what this looks like. So, on the right here is the 3 different colors of tracks represent 3 consecutive trips from 3 different individuals. You can see this red individual kind of seems to be visiting the same area consecutively, same departure angle, showing high level of foraging site fidelity. Then you have the yellow individual, which is kind of like the average level of foraging site fidelity that we would see. And you have this blue individual, which has kind of, there's no overlap, and it seems to be experiencing some kind of switching behavior. It's interesting to think potentially what decisions are leading to this switching. So yeah, I want to understand what is driving these patterns then. So we need to think about resource localization mechanisms that the gannet uses and stuff that Sam Cox has talked about as well, public and private information. So of course, gannets are visual predators. They kind of scan the ocean with their vision, looking for prey, and this is thought to kind of trigger area-restricted search movement. But they also have private information or memory effects, so whether that's accrued over kind of early life or just prior to the breeding season during incubation period when they have less constraints. And they also have a wealth of public information available to them. So even at Bass Rock with 150,000 gannets, even when you're potentially 100 kilometers away, you will still be encountering conspecifics, giving you potential cues to where food might be located. But how do these combine? We can start to kind of think of some potential hypotheses. But just first to kind of reiterate, the purpose of this study is to use simulation experiments to model different resource localization mechanisms, deriving public and or private information to decipher which conditions lead to the reproduction of empirically obtained foraging site fidelity patterns. So before I could delve into these kind of experiments and simulation experiments using different levels of social information and so on, I had to first kind of create a model in which it represented the movements of a typical foraging trip well. And we did this by using kind of lots of literature and parameters, previous models of gannets and different organisms as well, and our own empirical data, so lots of GPS data. And from this we withdrew step lengths, turning angles, to kind of inform how they move. This was done at 2-minute time steps in the model, which is key because that's how we collected our GPS data. So they're then directly comparable. We programmed a type 2 function response to basically as if they were foraging over areas which had higher density of prey, they were more likely to begin foraging. And it was based on this trap line strategy where you kind of gannets, you see linear trips and sometimes you see more looping trips. And it's thought that maybe they're kind of exploring on food but trying to limit their distance from the colony. We programmed in different movement modes that they could switch between, depending if they spot food, for example. So they begin at the nest, begin outbound travel, if they spot some food, then they begin area-restricted searching. If they're not successful, they'll return to outbound travel and kind of cycle on this until they hit a certain threshold, and then they'll begin inbound travel back to the colony. So to show you what that looked like, on the left is one empirical track, and on the right is a kind of very similar kind of type of simulated track that came out of that. And you can see that, they kind of have these foraging areas on the way out, and then they loop around, some foraging on the way back in, and inbound travel. So once we were happy with how movement was represented, kind of had average trips and also kind of the variation that we would see as well represented, we moved on to our simulation experiments. So the visual detection is already included, so now I'm just looking particularly at memory effects and public information. And these were done with 1,000 individuals each. Unfortunately, I couldn't include the whole squad. So first of all, I'd like to draw your attention to the public information. So we had different hypotheses here. We had no social information. So that was our null model. They wouldn't use any they would just ignore each other. Local enhancement, where individuals were attracted to others engaging in area-restricted search, so they fly towards them. We also include competition, thinking that why begin foraging in an area where there is lots of other birds? And then we had the kind of other one where they acted concurrently, where you would only begin or only uptake on an enhancement opportunity if you weren't experiencing competition. On the left, we have different levels of private information, the different hypotheses we developed there. We had no memory, again, our null model, where they would take a random departure angle each time from the colony. We had a long-term memory hypothesis where there was a burn-in period in the model, so they would do 10 exploratory trips, and they would pick which trip duration took the least time and remember this angle from the colony and depart on that. We also looked at short-term memory, where it's kind of a sliding window where they review the last 3 trips, and if they've been increasing in duration, then we will decide to pick a new angle. And then a combination of that, where we had a burn-in period, and they remembered 3 angles, the 3 most successful angles from departure angles from those 10 trips, and could cycle through them using their short-term memory, then this sliding window. And we want to understand which of these 16 different simulations then of kind of varying plausibility are most likely to reproduce patterns similar to our empirical data. And I can't show you them all, unfortunately. So I'll just focus on the null model and our most complex one here, which was, a spoiler alert, was the best at reproducing the foraging site fidelity patterns. So this is what I stared at for a couple of years. This is the interface of NetLogo, and I just want to show you one of the kind of models in action. So this is the burn-in period where they kind of go in on exploratory trips, and you can see the different colors represent the different movement modes. These trips will disappear once the burn-in period is over. The background, the prey, is informed from our own GPS data of over 500 trips of just foraging areas to infer prey density. So this is the beginning of the model then. It's decided I'm traveling northeast. That's a good direction to go in. But as the trips get longer, it's like, oh, maybe this area isn't proving fruitful anymore, so therefore head off on a different angle. So this is just one individual. We had it running a lot slower for more. So yeah, that kind of gives you an idea of what that looked like. And now I want to move on to the results. So first of all, looking at individual patterns. So this is the null model. Again, there was no social information or no memory being used to look at departure angle. And obviously, therefore, you see that within these colors, again, 3 consecutive trips from 3 different individuals in the different colors. You can see not much overlap within color, random departure angles. It doesn't really look like what we saw with the empirical data. Then this is for our most complex model. You can see there we have the red individual, again, shows that there was one showing very high foraging site fidelity. Then we have a kind of intermediate level, the yellow, and then this blue individual, which seems to be showing some kind of switching behavior. So we've captured, the way we quantify foraging site fidelity is kind of a bit of a snapshot. And that's the same way we've quantified it here, and we've obviously captured the switching behavior within that snapshot. But we also looked at some population level patterns. So what does this mean at population level? So these are the 50 and 95% kernels for 90 trips from 30 individuals from our null model in blue, overlaid on the gray, which is the prey density from the model. And you can see that they don't track this very well, especially in the kernel, especially out to the outer kind of reaches of the 95% kernel. Whereas for our most complex model, you can see that this has led to a much more informed population, both in terms of both kernels. They are exploiting the given area a lot better if they're using this private information to refine where they're traveling to, but also kind of social cues as well. So just to summarize then, flexibility in choosing between departure direction and interaction with individuals was key to reproducing foraging site fidelity patterns. I'm not saying that this is what the birds are thinking, but it's just a scoping exercise to kind of see and get an idea of potentially what different mechanisms might be at play. And this, interestingly, led to an adaptive population which exploited the given population well. So the outlook would hopefully be to be able to model management scenarios like offshore wind farms, and in a model where foraging site fidelity emerges naturally. You could look at things like how this might impact on trip metrics. You could look at body condition impacts, but also interestingly, which kind of emerged from the findings, was that you could predict the change in distributions of an adaptive population potentially, and maybe this kind of technique would work well. Complements some things like species distribution modeling, which I understand has kind of transferability issues. That was a very whirlwind tour of that. And I couldn't mention a lot of the results. So please come and talk to me if you'd like to know more about this particular model, or just if you want to talk about the technique in general. I find it pretty interesting. And I'd like to say, thanks to all these people. Thank you for listening, and happy to take any questions.
Thanks very much for an excellent talk. Very nice model in operation there, if complex. Any questions there?
Lunchtime.
Yeah. So in terms of the how testable do you think this sort of model is? Because it is a relatively complex series of things. How close are you planning to test some of the ideas in these?
Well, yeah, I mean, that would be nice. And I think as a technique, it has the potential, if you can tread the line of field work and then using that to inform models, and then using your models to inform field work, that would be a collection of empirical data. That would be great. But no, I don't have many ideas, because I think obviously the social influence is very important, and you could see from Sam's study that when you can tag a lot of the population, it's kind of easy to understand then what interaction is going on. Also, Terry Jones's work looking at a smaller population of gannets or a smaller colony, and you could kind of then sense lots of social interactions by tagging a lot of the colony at the same time. Obviously, that's not possible at Bass Rock. So maybe radar or something like that. Who knows? Yeah, I don't know.
I've just a question of detail about how those models work. So when they had conflicting information to deal with, be it private or be it social information, what was the general rule in terms of deciding how they would have weighed up the cost benefits of making or choosing which one won in terms of the decision?
So it was just they had to not be experiencing a certain threshold of competition. So an individual would assess how many people are in a certain radius around me, and if it was over a certain amount, they wouldn't even uptake on any enhancement opportunities. So they wouldn't be looking for other foraging birds. They would just be kind of getting out of there. And I don't know if that's plausible, but we kind of had to just come up with one hypothesis for each, really, and stick with that. I mean, obviously, you could have just done countless different iterations. So yeah, that was the way that particular hypothesis worked.
Thanks very much. I think we've probably just about run out of time. So I'd just like to ask you all to thank Chris, but also to thank everybody for, I think, an excellent range of talks on foraging ecology in this session. So thank you. We'll be back here